So we know that monogenic mutations in presenilin genes, in the APP gene, are sufficient to cause Alzheimer's disease in people who have the early onset form. But the question that needs to be answered is, what is the genetic basis for sporadic Alzheimer's? The kind that's typically late onset. And so the experiments that were done to at least somewhat elucidate or figure this problem out went like this. The first thing that scientists did was to immobilize, oops, A beta, right, this fragment, on a special membrane. The idea here is you've got some special membrane and A beta just sticks it there. It doesn't move, it doesn't degrade, it just sticks. Okay, so it's immobilized on a special membrane. And then the goal was to see what proteins from Alzheimer's CSF bound. So see what proteins associated with this A beta. So see what proteins. Now, what do they get these proteins? From the cerebrospinal fluid of Alzheimer's patients. Right, so the idea was we want to know what proteins exist to interact with A beta, and they're likely found in the circulating fluid, the cerebrospinal fluid that sort of sloshes around the brain and spinal cord, and to determine if there's anything in that fluid, any of those proteins, that would complex together with A beta. And if they do, the thought is that maybe they work together, right? And so the result of these experiments was a protein called apolipoprotein E, which we'll just call ApoE, did indeed associate with A beta. Okay, now normally ApoE, if you've talked maybe about cholesterol movement through the blood, you've talked about apolipoprotein E before, this is a molecular chaperone for cholesterol, right? It helps to shepherd cholesterol or move cholesterol around the bloodstream and get it where it needs to go. So a chaperone for cholesterol. In the blood, right? So the idea is it's a protein, but it's associating with lipid, right? Apolipoprotein, okay? And it turns out that a separate discovery was made that the gene encoding ApoE was on the expected chromosome, like we talked about before. All right, so this, I think, led people to believe that probably ApoE did have something to do with Alzheimer's. Well, the question is what? Well, ApoE has 3 isoforms. Right, so you know a little bit about isoforms where you have a gene that can get it alternatively spliced, and you basically get the same functional protein, but slightly different sequences. ApoE2, E3, and E4. Okay, and it turns out that in late onset Alzheimer's, the E4 allele is way higher, right? It's like 4 times higher than the E3 allele of the general population. than the much more common E3 allele. Oops. Okay. Additionally, people who are homozygous for E4, right? So they have 2 copies of E4. Right, those that are homozygous are 8 times more likely to develop AD. Okay, now it's really important to note that this E4 allele is not causative. So E4 allele does not cause AD, right? It's just a risk factor that if you have it or you have multiple copies of it, that you're more likely to get it. Whereas the mutations of APP or of the presenilin genes, that was sufficient to trigger Alzheimer's. This was not. This just shows an increased risk factor. Okay, so let's talk a little bit about cellular pathology now, right? What we think happens, like I mentioned before, is that APP processing is changed in some way, and that the A beta accumulates. So let's look at that next. So what do we need to know here? We need to know in this diagram that APP is a transmembrane protein, right? So it's sticking in the cell's membrane. Transmembrane protein. Okay. It's cleaved, right? So it's able to be cut. What are called secretase enzymes. And different products have different solubilities, okay? And so things that are soluble are able to be broken down by the cells, and things that are insoluble can't, and they tend to aggregate. So the soluble cleavage products get broken down, whereas those insoluble products can't. All right, so let's look at this pathway that's working quote-unquote normally, right? When APP, which is always present, right? Your cells in your brain have APP. It doesn't mean that you're going to get Alzheimer's. Why? Everybody has APP. So let's focus on this side of the pathway first with this protein APP that, again, it's present in everyone. So maybe we should note that this is the non-AD path, because we want to serve to contrast this with the Alzheimer's developing pathway, right? So in this pathway, APP is cleaved by alpha secretase. Whoops. Into 2 different products, a soluble APP alpha as well as something called CTF alpha. CTF is the C-terminal fragment. Okay, so this is a protein, right? And so you have, you've got the this is all the protein. There's some alpha helices here, beta sheets, and whatever, right? And so it's inside the cell, right? This is the C-terminal end, right? The carboxyl end, and there's also an N-terminal, right? This is any protein has an N-terminal and a C-terminal, and it just so happens that this C-terminal end is sticking out. And so cleavage via alpha secretase produces the soluble form of APP, right, the fragment, as well as the C-terminal fragment alpha, so named alpha because it's cut by alpha secretase. A second secretase then cleaves CTFA, or CTF alpha, I should say. Gamma secretase. And the result of the cleavage by gamma secretase is this segment of AICD. Oops. APP, intracellular C-terminal domain fragment. Okay, so this is just a peptide, right? This is just a peptide. These are just, shorter chains of amino acids than what we started with. And because, we'll focus on this thing, right? This is the soluble portion. It can be broken down. This AICD actually has some role in downstream signaling. And so we won't go into that too much. None of this is particularly harmful. In the plaque-forming pathway, APP is cleaved first by beta secretase. Right? And this forms a smaller soluble region, right? SAPP beta, right? Soluble APP beta. And so we're not going to worry too much about this. But because it cuts at a different region, right, than the alpha secretase did, the product that we get, the CTF beta, is different. Right, the C-terminal fragment of when it's cut by the beta secretase is a little bit longer, right? So this was the CTF alpha, and you can see that it's smaller, it's shorter, fewer amino acids than the CTF beta. All right. Well, then what happens is this CTF beta is cleaved by gamma secretase, but this time what's left is a little bit different besides the AICD, right? So the AICD still forms, right? That is the APP intracellular C-terminal domain, right? Still forms. But the product of both beta and gamma secretase action is this segment right here. This is the A beta. Okay, and this is insoluble. And it tends to aggregate. It tends to stick together and form these long fibers. And these long fibers aggregate together to form plaques. Okay, so you can see you've got a fiber here, and a fiber here, and a fiber here, and then they stick together, and now we have these plaques that form. Okay, and so what does presenilin do in all of this? Right, well, you can see that presenilin is here. The mutation changes how the gamma secretase cuts, or the gamma secretase cutting activity. And so we think that presenilins regulate the activity of secretases. Now, this is oversimplified, right? So they show a secretase, right? And a secretase is made up of presenilin, but we know there's presenilin 1 and presenilin 2. There's also other proteins in this cutting complex. Okay, so it's complicated, and different mutations bias different secretases to cut differently, right? So if we were to somehow downregulate the activity of gamma secretase or upregulate the activity of alpha secretase, that could be a potential treatment for Alzheimer's. So could a drug that prevents plaque aggregation. If this A beta was produced but didn't form plaques, maybe that's a potential therapeutic. Drugs that break down these plaques, right, that can dissolve the plaques once they form, are potential therapeutics, right? But what I want you to take from this is that APP is a really common, right? Your cells, your neurons have amyloid precursor protein, and in the normal, quote unquote, normal non-pathogenic role or route, secretases still cut, but they cut APPs at different sites. Those products tend to be soluble and not give us much trouble. But if the secretases cut differentially, if presenilin is mutated, they do this, right? So in mutated presenilins, we know that gamma secretase is overactive. That leads to more A beta formation and these plaques, right? So both things, we think that both pathways are happening in sort of in healthy brains. But if this one's happening to a much lesser degree than this one, which we think of as the quote-unquote normal pathway, this person doesn't develop Alzheimer's. But right, over time, if this pathway becomes increased and this pathway is downregulated, that may lead to Alzheimer's. Okay, so that's what we'll say about Alzheimer's. A lot yet to be understood about it. We don't know what the APP protein does normally. We don't necessarily understand how the plaques trigger cell death. We think that it has something to do with interrupting intracellular transport, right? That you have these big aggregations and now things can't be transported throughout the cell. But much of this is yet to be known, and there's a tremendous amount of study that's going into it right now. I think what we'll do is come back and maybe we'll talk about, we'll lay the framework for Parkinson's disease with the rest of the lectures today.